Welcome to our video lecture on carbohydrates and lipids. And of course, if we're talking about carbs and lipids, we've got to have some snacks, right? Our essential understanding for today is that compounds of carbon, hydrogen, and oxygen are used to supply and store energy. Remember that carbs and lipids are both composed of carbon, hydrogen, and oxygen. Objectives for today, we are going to name examples of monomers, dimers, and polymers of carbohydrates. We're going to talk about some functions of those carbohydrates. We're going to look at the differences between straight and branched chains of polysaccharides. We're going to state some examples of carbohydrates that are composed of alpha glucose and some that are composed of beta glucose. We're going to name examples of lipids and their functions. We're going to talk about the differences between saturated and unsaturated fatty acids, as well as between cis and trans unsaturated fatty acids. And then we're going to wrap up with a little look at BMI, body mass index, and how it can help predict some health concerns. Again, our essential understanding is that compounds of carbon, hydrogen, and oxygen are used to supply and store energy. So that's the biggest function of carbs and lipids. Interestingly, lipids contain more energy per gram than do carbohydrates. So we can store about 9 calories of energy in every 1 gram of lipid, only 4 calories per gram in carbohydrate. An advantage, though, of carbohydrate, they are soluble in water, so they're easier to transport around the body in the blood. Lipids are not soluble in water, so they need a little bit more special transportation. We can use carbs in both aerobic and anaerobic respiration, whereas our lipids are only useful in aerobic respiration. So lots of vocabulary here related to our carbohydrates. Remember that monomer, polymer. Monomers are those one tiny little pieces. Polymers are chains of lots of monomers bound together with those condensation reactions. When we're talking about carbohydrates, sometimes instead of using monomer, we use monosaccharide. And so this is just a special kind of monomer of a carbohydrate. You need to memorize some examples of monosaccharides. So IB wants us to know that examples of monosaccharides include glucose, fructose, ribose, and deoxyribose, and then galactose. So these are some important monosaccharides for us to memorize. We can take some monosaccharides and bind them together into disaccharides. Disaccharides are special dimers of carbohydrates. And the three that we need to know are maltose, sucrose, and lactose. Maltose is a dimer composed of 2 glucose monomers. So this is maltose. It's glucose-glucose. We're going to talk in a couple more slides about alpha and beta glucose. All of these guys on this page are all alpha glucose. So alpha glucose to make maltose. Sucrose is composed of alpha glucose and fructose. Sucrose is here, glucose, fructose. And then lactose is composed of glucose and galactose. Lactose is glucose and galactose. Memorize these. All right, so remember that condensation hydrolysis reactions. If I'm taking 2 alpha glucose monomers and I'm putting them together to make this dimer, maltose, a disaccharide, it's going to be a condensation reaction, and this is an example of anabolism. Just a little bit of review from our previous lecture. So I mentioned this idea of alpha glucose. There's also a beta glucose, and the difference is simply this. So this is carbon number 1. We call this carbon number 1. And on alpha glucose, the OH group, the hydroxyl group, points down, whereas in our beta glucose, that hydroxyl group points up. So we've got carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6 in our glucose ring. Oxygen is that other little point of the hexagon. Hydroxyl down, down, up, down. Whereas in beta glucose, we have oxygen, carbon number 1, up, down, up, down. Alpha glucose, beta glucose. Alpha glucose will bind together to form what we call glycosidic bonds. Anytime we put monomers of carbohydrates together, we form glycosidic bonds. And what's going to happen is, with our 2 alpha glucose guys, we're going to take this and this and they're going to come together in that condensation reaction to make water. So the OH of one and the H of the other. That leaves this oxygen missing a bond. It's going to stick to this carbon number 1 on the next carbohydrate. This is called a glycosidic bond. Carbohydrates form glycosidic bonds between their monomers. And if I have alpha glucose, I can make all. So we're going to look at some polysaccharides. These are going to be polymers of carbohydrates that are formed from alpha glucose. These are going to be our middle length energy storage. So longer than our monosaccharides or disaccharides, but not as long-term energy storage as our lipids. Plants make starch and animals make glycogen. So starch and glycogen are both composed of oodles and oodles and oodles of alpha glucose monomers bound together by those glycosidic bonds. So what's interesting about starch, it's actually composed of two components, amylose and amylopectin. Amylose and amylopectin. Amylose has what we call only 1,4 glycosidic bonds. Glycosidic bonds are the bonds that form in polymers of carbohydrates. In amylose, we have only 1,4. And when we talk about these 1,4 bonds, because this is basically organic chemistry, we're counting our carbons. We love to count our carbons in organic chemistry. So if I'm looking at a monomer of glucose, and I'm not drawing all the hydrogens or even all the hydroxyl groups, I'm really just putting those important carbons in there. I have carbon number 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. We love to count our carbons. When I'm making amylose, I'm going to bond carbon number 4 to the next guy's carbon number 1. So I'm going to add another carbon over here. So there's our carbon number 1, 2, 3, 4, 5, carbon number 6, and then our oxygen there to finish out the points of the hexagon. So what's going to happen is we're going to form a bond between carbon number 4 and carbon number 1. This is our 1,4 glycosidic bond between alpha glucose monomers. Glycosidic bond, carbon number 1, carbon number 4. This is what we have, the only thing that we have in amylose. And so amylose is going to form what we call a straight chain. And it does do this spirally thing, but there aren't any branches on our bonds. That's different in amylopectin. So what we do in amylopectin is we add another bond on another carbon. We're going to now add carbon number 6. So I'm going to take carbon number 1, which is now kind of down here on the bottom. Good, so here's carbon number 1, 2, 3, 4, 5, 6. So another alpha glucose monomer, I just twisted it around a little bit. And now we're going to form a bond between carbon number 1 and carbon number 6. So you can see instead of having straight chain like this in amylose, amylopectin is going to branch off in different directions. And so here we have 1, 4, 1, 4, 1, 4, 1, 4, 1, 4, and now we're adding that 1, 6, which is going to make branches. So amylose, straight chain, and sometimes it curls up like that, but it's still just going 1, 4, 1, 4, 1, 4. That's different from amylopectin. So here's 1, 4, 1, 4, 1, 4, 1, 6, 1, 6, 1, 4, 1, 4, 1, 4, 1, 6. And so we're going to have more branches in our amylopectin. Amylopectin and amylose together make starch. It's all alpha glucose. It's all glycosidic bonding. It's just a matter of, is it straight chain 1, 4 only, or does it have branches 1, 4 and 1, 6? Glycogen is the animal version of starch. It is 1, 4 and 1, 6 because look at all those branches. 1, 4, 1, 4, 1, 4, 1, 6, 1, 6, 1, 6, 1, 6, 1, 6. Glycogen is actually far more branched than is amylopectin. So even more of those 1, 6 bonds in glycogen than in amylopectin. This is a giant molecule of glycogen, and you can see all those branches, but each one of those little divots, each one of those is alpha glucose, alpha glucose, alpha glucose. What's kind of nice about us binding together all of this glucose like this is that I can roll this giant molecule around my body, and when I need to make some ATP, we can just start chopping off all those glucose molecules, send them to our mitochondria, and make lots of ATP. So now we're going to flip over to this idea of beta glucose, different from alpha glucose. Beta glucose, again, just that hydroxyl group going up instead of going down, is what makes up cellulose. So we're still going to have glycosidic bonds because I'm still putting together monomers of carbohydrates. But now instead of making starch or glycogen, we're going to make some cellulose. Cellulose is composed of beta glucose, not alpha glucose. What's interesting about beta glucose, because this OH going up and here, it's harder for us to make the bonds between the adjacent glucose molecules. And so what we have to do is instead flip one of those guys upside down so that these guys kind of line up, and then I can steal that H and I can put that OH, condensation reaction. We're going to make that water, and then this oxygen will bond to that carbon number 1. So I still have my 1,4 glycosidic bonds, but now I had to flip one of the glucoses upside down, and so we're going to kind of alternate an up, a down, an up, a down as I put together my chain of beta glucose molecules in cellulose. So we have a down and an up and a down and an up, and this results in super cool forces of attraction between different layers of cellulose. So I can have this one chain, and because of this oxygen up here and this oxygen down here and this oxygen up here, we can set up some hydrogen bonds, and these fibers of cellulose, so this is like beta glucose, beta glucose, beta glucose all along here, held together by our glycosidic bonds. They're going to have these super cool forces of attraction, electrostatic forces of attraction, like little magnets where these fibers of cellulose stick together. And then we have lots of these fibers will make up cell walls of plants. What is super cool about these cell walls of plants is that we mammals do not make enzymes that can break down this weird bond because of the up, down, up, down, up, down. So our enzymes don't quite work, which means that cellulose for us is fiber, which is pretty cool. All right, so now we're going to flip away from carbohydrates and look at lipids instead. So when we talk about lipids, we don't really have monomers and polymers. They're just kind of all big molecules. The one exception might be this idea of fatty acids. So we can put multiple fatty acids into the same molecule. It's not like a chain like our carbohydrates, but more just like little clumps. But fatty acids kind of are monomers, but not really. But fatty acids are going to be crazy long hydrocarbon chains. Hydrocarbon chains just mean there's hydrogen and carbon only. So you can see carbon, 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 all of the hydrogens here. Remember that valence of carbon is 4, so 1, 2, 3, 4 bonds on every carbon. And the very end of a fatty acid is going to have this carboxylic acid group. Remember that there's one of these also on amino acids. This is the part that makes the fatty acid the acid. This is the part that makes it fatty. So that carboxylic acid group, we're going to have this, the final C is going to be double bonded to an oxygen and then also to this OH group. We call that the hydroxyl group. This is a carbonyl group, and when I put a carbonyl, double bonded O, and hydroxyl OH together, we end up with a carboxyl group, also known as a carboxylic acid group. So fatty acid chain, crazy long chain of carbons, and then at the very end, this carboxylic acid group, C-O-O-H. Good? Good. All right. So our fatty acids can be saturated or unsaturated, and unsaturated can come in a couple different forms, mono or poly. Mono means one, poly means more than one, right? So saturated fatty acids have no double bonds between their carbon atoms. No double bonds. They're going to be single, 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 single. All these bonds are single bonds. Remember that carbon has a valence of 4. So each one of these carbons is bound to 2 carbons and then also 2 hydrogens to get to those 4 bonds. If I throw a double bond into my carbon chain, so I put a double bond between two carbons, now this carbon has 1, 2, 3 bonds with carbon. It only needs one H instead of 2. So we call these guys saturated because they are as full of hydrogens as possible. We have the max number of hydrogens possible. This is an unsaturated fatty acid because there are fewer than the max possible numbers of hydrogens. We actually end up with these little holes where those hydrogens have been removed because that double bond took their place. If I have more than one of these double bonds, I have a polyunsaturated fatty acid. What's kind of cool about our fatty acids that are saturated, unsaturated, saturated fats, you probably know are bad for us. These tend to be solids at room temperature. We consider these more of our fats. These fatty acid chains that are unsaturated, either mono or poly, tend to be liquid at room temperature, and so we tend to call these our oils. So oleic acid in olive oil, stearic acid in butter. So just beware. Saturated versus unsaturated fatty acids do have some different health risks involved in their consumption. Unsaturated is definitely better for us. But not all unsaturated fatty acids are made the same. So we have two different kinds of fatty acids that are unsaturated. There's cis and there's trans. Cis means same and trans is opposite. So this cis unsaturated fatty acid simply means that those hydrogens that we're keeping on the double bond are on the same side. And the hydrogens that we took away because we made the double bond instead are on the same side. And this opens up this giant little hole in the molecule, which allows it to bend a little bit. And this bend, or this kink in the molecule, means that it cannot pack together as much. It's going to have a lower melting point. These oils are better for us. Our trans unsaturated fatty acids, there's still that double bond. We're still missing some hydrogens, but we put those hydrogens on opposite sides of the double bond, which means that they kind of balance each other out, and there's not that big gap that allows for a big bend in the molecule. So this is more straight-ish, which means that these guys can pack together more. They're more likely to be a solid at room temperature, and this is where we get our amazing margarine. And so even though these guys are like, hey, we're made from olive oil, well, but we made the olive oil a trans unsaturated fatty acid, and these are not as good for us as are our cis fatty acids. Good? Good. All right, so now we're going to look at some of our bigger molecules of lipids, how we can put together some fatty acids to make what we call a triglyceride. Tri is for 3. Glyceride is because we're starting with this molecule, glycerol. And I'm going to add to the glycerol 1, 2, 3 fatty acids. Again, tri for 3. So glycerol has 1, 2, 3 OH groups, these hydroxyl groups, and I can take the hydroxyl group from the carboxylic acid group of each of the fatty acids and bond these together and make some water in our condensation reaction. So I'm going to take the OH and the H, and that's going to make water. This O is then going to bond to that carbon, and this kind of bond is called an ester bond, and that's what we see right here. This is an ester bond, and I can do that to each of these guys. So add a hydroxyl group to each one of those fatty acid chains, and then I have my triglyceride. And because I had condensation reaction here and here and here, we end up with 3 molecules of water being formed when I remove them, when we take off the hydroxyl group of one guy and the H of the other guy to make those ester bonds. So triglycerides, 1, 2, 3 fatty acids on a glycerol molecule. What's kind of cool is that these fatty acid chains can be saturated or unsaturated or polyunsaturated or a mixture of all 3. So now let's look at some lipids that are not based on fatty acid chains. We have some waxes like what leaves what plants put on their leaves to protect them from dehydration. Beeswax is also a lipid that's super cool. We have lots of sterols. These are hormones that are based on the molecule cholesterol. So sterols formed from cholesterol. So you can see here, estrogen, progesterone estrogen and testosterone look a lot alike. Progesterone also looks similar. These all look similar to cholesterol, are cholesterol based. We have some fat-soluble vitamins. These are all made of lipids and are super important for health. And then, of course, we have our lovely phospholipids that make up our cell membranes. A little bit more vocabulary here. So adipose tissue is fat tissue. It's our fancy scientific name for fat. What's great about adipose tissue is that it's easy to carry because we can put so many more calories. Remember, 9 calories for every 1 gram of fat. We can throw a whole lot of energy into those fat storage molecules. So for birds and bats that have to fly, the amount of energy needed to fly is huge. It's super easy for them to carry around this fat. It's not dense, lightweight with lots of energy. Adipose tissue is also amazing for heat insulation. And so especially our aquatic mammals that live at the poles, whales and seals, will have huge amounts of adipose blubber layers to help with heat insulation. And then for us, we also use adipose tissue as shock absorption. So our hearts have a little bit of fat around the outside. Fat on the outside of your heart is good because your heart is right behind your breastbone. And you can imagine that if your heart knocked into that bone every beat that it beats, that would build up some scar tissue. The layer of fat around the outside of the heart is really good because it kind of protects your heart from knocking into that bone. Of course, if you start getting fat inside your heart, that's less good. Which brings us to the idea of BMI, body mass index. So we calculate BMI by dividing mass in kilograms by height in meters. It has to be kilograms in meters for IB because IB is all about the metric system. And so then we can compare this ratio of mass to height to a BMI chart. Underweight is here in blue, healthy is here in green, and then we get to overweight and obese and extremely obese. And again, this is by comparing weight in kilograms to height in meters. I know this says centimeters, but I also added in this BMI chart, we have English standard measurements if you want to check your own BMI. If you have a very high BMI, if you're starting to head toward overweight or obese, then you are at increased risk for coronary heart disease, type 2 diabetes. Your life expectancy might be reduced because of the diabetes, because of the risk of heart attack or stroke. And then you also tend to have an increased cost to stay healthy. You have more medical bills, you need more medicine, you need more stuff to keep you alive. And so it is super important to try to keep your BMI at a healthy place. A little bit of a caveat here. Of course, some of us are just denser than other people. Some of us are bonier than other people. And so even though BMI is a great predictor, it is not the be-all end-all of are you healthy or are you not healthy. But if your BMI does start to creep up or down, being underweight is also unhealthy, this is a good check to go look for other things and see what other risk factors you might have. How do we calculate BMI not with a calculator? So we have these super cool things that are called nomograms, and you literally just find what is your body mass. Maybe your mass is 60 kilograms, and then you go over here to your height and you're like, I am 145 centimeters tall, and then you draw a line between those two, and wherever you cross in the middle, it looks like the BMI of this random person is, I don't know, 27-ish. And so you can calculate BMI also with this nomogram just by connecting dots. And that, my friends, is the end of our lecture for today. Did we accomplish our objectives? Yes, we did. We named some examples of monomers, glucose, fructose, galactose. We talked about our dimers, maltose, sucrose, lactose, polymers, our amylose, amylopectin, and glycogen, and cellulose. We talked about some functions of carbohydrates, our short and middle term energy storage. We understood straight and branched glycosidic bonding. Straight chains are 1,4, branched is 1,4 and 1,6, glycosidic bonding in polysaccharides. We talked about alpha glucose and beta glucose. Beta glucose is mostly cellulose, which is more of a structural carbohydrate in our plants. Alpha glucose tends to be more for energy storage. So this is our sucrose and our glycogen, our starch molecules. And then we flipped over to lipids and we talked about some examples of lipids, those fatty acid chains. We looked at saturated and unsaturated fatty acids. We talked about cis on the same and trans on opposite side, unsaturated fatty acids. And then we wrapped up with BMI, body mass index, and how it can help predict but not be the be-all and end-all of health concerns. Good work today, my friends.